Okay, okay. Daniel, please.
So hang on. Okay. Hi, everyone. So as Eve said, my name is Daniel. I'm currently a postdoc at Utah State University, but I was here in Panama starting 2021, more or less. I was supposed to come here during the pandemic, but it was a strange year for all. But since then, like I've been luckily involved with Eve and with the amazing work that they do here. I joke with people that I am actually a fake entomologist because I don't know. And the more I learn about it, the more I realize that I have no idea about anything. So I've been really lucky to be actually involved with the metabarcoding project, but I will go a little bit more into detail about this. How can I? Oh, hang on. I think it's a problem. Yeah, I got it. Okay. So just like a few, I think, interesting facts about insects or diversity of life in general. There's about 1.4 million described animal species, and the estimations are completely wild. Yeah. So there's about 5.4 million species ready to be described. E.O. Wilson estimated like around 10 million species in total. Then other people got really excited with this idea and they started doing their own estimations. And so Eve mentioned one recent estimation about Hymenoptera overtaking Coleoptera. And that's because they have these models showing that, okay, if each species has a parasite, which is in the Hymenoptera, then for every insect species, there will be one parasite that more or less like duplicates everything, right? And I like, okay, it's a old reference by Carvalho and Marques. These are Brazilian researchers. So the costs are probably much higher today, but they estimated how much it costs to actually describe one Brazilian vertebrate species. And this considers, the cost of fieldwork and hiring researchers and assistants and going into the field, finding the animal, taking it to the museum, finding some like decrepit taxonomists to actually describe the species. So it's about $122,000 for one new vertebrate species. The good news is that insects are relatively cheaper. It's only almost $40,000 per species. And so if we calculate for those 5.4 million species waiting to be described, it's about $263 billion, which I don't think there is money in the world for that. And it would take 360 years to actually complete. And I will not go into funding. I think we already have some, depressing stories with that. But what is interesting, and we've all heard about this recently, is the insect apocalypse and the insect diversity is crashing, and we're bracing ourselves to a catastrophe. Some, I think, serious researchers are thinking that we're entering the 6th mass extinction. The previous extinctions were all, due to planet history, and the 6th mass extinction may be caused by us. And even if we're not entering, they have some doubt of how things will go, right? But even if we're not entering a mass extinction, we do have problems with, of course, forest loss, land use changes, pollution, agriculture, and a lot of issues that are preventing us from knowing what really is out in the world. And as I just mentioned before, we probably have no idea of how much there actually is. So, because taxonomies, it's a very slow process and it's a very hard work. Luckily, DNA sequencing technology has allowed us to accelerate this a little bit, especially now that we have idea of cryptic species, and by morphology alone, you cannot tell the differences. So, okay, we have these pieces of DNA that we can sequence, and there will be some difference between the two species, right? The cost per genome is decreasing, although we were discussing this earlier, we still pay the same for sequencing, but apparently it's getting cheaper anyway. DNA barcoding. So this was a very interesting idea, a few years ago, that each living thing, we have DNA, of course, but there are some segments of DNA that are shared between the groups. And there are short regions, which are highly variable between species, but quite conserved within species, right? So I don't know if people here also work with plants. We probably do. It's a little bit harder there because the single gene is not enough to actually tell you the species, but you can use different markers. And I am sure here in BCI, they have used ITS and RBCL to do like the barcoding of the island. For animals, it's CO1, the cytochrome oxidase 1. Fungi, it's ITS. And then bacteria have a different one. But there are certain genes that we can use to identify species, right? And the way we identify these species is by this thing called the barcode gap. So you take that little sequence of DNA, and then you have, ideally, multiple individuals of the same species, and there is some difference, just natural variation in that piece of DNA. And between species, that difference should be higher. So you can calculate what's the intra-species variation and the interspecies variation. This is the barcode gap. And there is a lot of debate about this. We're not exactly sure, or there is no direct cutoff, but somewhere between 2% and 3% and 4% difference for animals in CO1, it's supposed to be enough. And some of this is basic, so I hope I'm not boring you yet. But there is a very great example with this Astaptes fulgerator, which kind of set the stage for barcoding in general. And it's a great paper. It's 10 species in one, because by morphology alone, they were unable to identify these different, what they thought was a single species. You also had to look at the genitalia, and even there it was very difficult to tell them apart. You need like super specialized knowledge. So what Paul Eber and others did is that they took larvae from the host plant, and they tried to get all these what they consider to be differences between these morphotypes. And finally, with CO1 sequencing, they were able to tell them apart. And there are actually 10 different species rather than one. And when they do the phylogeny of CO1, they all fall into different clades. And then, okay, a lot of studies after that came. They actually have different host plant species, and they can feed, and they're specialized, and etc. All these sequences become publicly available for public use. And this has like unleashed, well, I think it used to, now it's a bit slowing down, but the number of barcodes that people are actually now generating. Yeah, people go out into the field, collect whatever they can. We try to make barcodes, and there is public databases, some specialized for fungi, for birds, but I will spend most time talking about Barcode of Life because it's the one that historically Eve has used, and it's the repository with the most animal CO1 sequences. And I really like their mission. Yeah, it's to illuminate the biodiversity by developing globally accessible DNA-based systems to speed species discovery, probe interactions, and establish biodiversity baselines. One of the biggest criticisms with all these insect apocalypse studies is that, yeah, okay, there is a huge decrease right now, but we don't know what was there before. Like these studies were, I don't know, sometimes 20, 30, 50 years, but before then, we have very little idea. So you can see a collapsing trend, but you don't know if we are reaching the baseline or if we're past the baseline. So with this kind of studies and repositories, we can actually start to build a little bit an idea of what there is in the world. So at the moment, they have like millions of sequences, about 1 million beans. We will discuss at length the issue with genes and the problems of if they actually match or not. So thank you for the question. But the Barcode of Life now has about 1 million sequences. The great thing about this repository compared to others, for example, GenBank in NCBI, it's a bit restrictive for the regular user because they require you to have a picture, to have a description, to have like very detailed information about where that sequence comes from. I don't know if any of you have experience uploading sequences to GenBank specifically. I do. And I just send the sequence, but I am nobody to actually identify the thing I send the sequence from. I just have an idea and I think, okay, it is this species, but I don't have to upload a picture. There are some checks that they do to make sure that it is what I say it is, but there's a lot more variation. And one success of the Barcode of Life system is that although it's really restrictive of how you can send a museum quality sequence, it's actually useful then for the broader audience. Not to say that GenBank isn't. So now we have these gigantic reference libraries. And before you had to get one individual, sequence the gene and send it to them. But now with the high throughput sequencing, you can actually produce millions of reads or millions of sequences that then you can compare to these available databases, right? That's also how you build genomes, yeah, but it's the same individual. You take millions of pieces of that genome and assemble it together and you have a full genome. But if you actually send mixes of, in our case, insects, then you will start randomly generating sequences for everything that was in that insect soup. And then because you have a reference library, you're able to match those sequences to what we think are species, right? So this is like more or less trying to explain how metabarcoding works. I am sorry if this is too basic for you. As I say, there is tons of repositories for some are targeted for bacteria, some are targeted for fungi, some are for like barcode of life for most animals. And then there is, I like the title of this paper because it's a pile of pipelines. There are almost as many pipelines for analyzing metabarcoding data as there are researchers doing metabarcoding. And you have a lot of control and liberty of which parameters you use, how similar you allow the sequence to be to other sequences. And it depends a little bit on your question. Like we here were interested in arthropod species, but some other researchers may be just interested in like a general community of what is there. They don't need to know necessarily each species. Some people don't even care about the species and they will just go for what they call exact sequence variants or OTUs, which are the operational taxonomic units. So there is a lot. This is just to summarize that. Again, going back to BOLD, is that you actually have a lot of information. If you find the same species, it has been collected elsewhere, and like you have a lot more metadata for that sequence that you generated, right? And as Yves said, it's 100 gigabytes of data from this project alone. So the data management becomes something to consider. So now I'm going to get into part of our actual project. When I came here in 2021, Greg had funding for like a pilot study for metabarcoding using light traps. So this was, let's say, like a preliminary experiment before the bulk of the Senaseed data would come. So Yves already explained the BCI. It's interesting. It's like a forest that is at least 100 years that we know has been preserved and maintained. And it's a lovely place to work. The arthropod monitoring team has been doing this like yearly census, 4 census per year for the last 16 years. The thing with long-term data, which I think is kind of a shame because you start building your data, then you get into 10 years of data, and then you can start doing time series. So the 9 years before then are a little bit difficult in terms of publication and what you're actually finding. But now we can actually start to tell stories about the trends of the insects on the island. And yeah, so the example of the more winners and losers, you've already explained. But there's also about Euglossa. So I don't know how many years that is, but many years, 40 years of data on Euglossa. So before the monitoring, but they have comparable data. And there's almost no change in Euglossa, which I think is kind of interesting. It doesn't mean it's the same case for every insect species or for every guild or for every group. But now you have the baseline, or we pretend to. On top of that, Yves has already discussed, but for me, the most interesting thing to talk about right now is that for BCI, we have, I think this number has been updated, sorry, this is a bit old, but about 13,000 sequences from 11,000 of individuals that have been sequenced. So we have a very strong reference library just for the island, okay? And so these were like our first attempts at learning how to generate metabarcoding data, learning how to use it. So this study looks at the ants, collembola, and termites. So in this case, we had parallel samples, one that was going to be sorted. So we separated, not we, but they separated collembola, ants, and termites for one trap, all metabarcoded, and other trap for manual identification. And so the first thing is to see, okay, with our reference data sets, are we capturing the same diversity using both methods? I think this is one of the most important questions in metabarcoding, because you need to know that you're actually. So here in orange is the metabarcoding data. In purple is the manual sorting. And we can see that, okay, for termites, metabarcoding was a lot better at capturing diversity. But for ants and Collembola, okay, some branches were better with metabarcoding, some branches were better by hand. But we have like a more or less matching situation of the known species of the island and what we collected with metabarcoding. This is all in the same plots that Yves has described. So where the Senaseed project has been done is the place where they have done the monitoring, and it's the place where we do this, I want to say pilot, but it was mega study anyway. It was only 10 light traps instead of the 25. 2 non-consecutive nights. Because of the pandemic, yeah, we were not able to actually sample in 2020, but it's okay. So then we had like 40 samples for the metabarcoding and 40 for manual classification, right? So this is just taking advantage of what the monitoring team already does with the light traps, but at the same time, we used metabarcoding, right? What we did at first was, to collect all the catch. Then using a simple handheld blender, we turned from this to like the classic insect soup. This was in 2019. We changed the protocol later because once you blended your insects, you have no chance of ever knowing what they were, right? So if you suddenly describe a whole new genus, okay, you don't have the animal anymore. You have just the soup. So the next time we actually sorted by size and kept the bigger animals out, just put a leg in there. And then, okay, we never actually sorted them again, but they are in storage. And if something incredibly exciting comes out of those results, you can at least have the chance to look at them again. So, okay, these are just parameters of the mBrave platform. mBrave platform is one of those piles of pipelines that you can use to analyze your metabarcoding data. This one, it has a graphic user interface. So I think that is very helpful for a lot of us that are not proficient in bioinformatics or with coding in general. And it has comparable settings to all those like custom-made pipelines. And we can talk about the actual parameters if you want, but just what you need to know is that we control for quality of each read. We trim the read to be a specific length. And then that read or that cluster of reads, because then the program will take all your sequences, hundreds of thousands of sequences, and make clusters of them and match it to the mBrave reference libraries, which are all part of BOLD, the Barcode of Life system. And one, I think, unique thing of mBrave compared to other pipelines is that you can actually give it the order of data sets that you want to use to classify your sequences. So in this case, we have an excellent arthropods from BCI dataset. So we use that as the first reference. Every sequence that matches to that reference dataset will not be searched in the subsequent databases. So you have a giant sample size or pool of sequences that gets reduced every time as you go along through the references of mBrave. So control insecta is just every single BOLD reference sequence that is available. And this is how we come to the bin, the barcode index number. It's a sequence, a representative sequence of those clusters that are already described. And I think Jeremy will talk more about these references and how you can build them, because I will get into it later. So now we have the huge light trap dataset. We were able to identify 4,611 individual species just from 40 light trap samples in 2 seasons, which I think is amazing. But we can talk about how amazing it is after. Then you can actually, because we have this refined level of information of species, you can also start looking at differences between seasons. I know this is not groundbreaking results, right? Oh, the wet season and the dry season have a different community. It's not groundbreaking, but it's groundbreaking when you consider it's 4,000 species that we're looking at, yeah? Most ecological studies will have a few hundreds, maybe a few thousands, but now you can actually do this mega analysis. We can look again at the distribution of different orders in our samples, and each dot represents one plot. So you can actually start looking at spatial differences based on metabarcoding data. I mentioned that I'm fake entomologist because I can't identify a single species, but I can look at bins and I can start telling about the composition of plots in the forest. We wanted to compare it with the data set from the manual monitoring program. And we can see that there is significant, in purple is the parallel sample. So the light trap that was collected at the same time and sorted manually. There is, I think, a good amount of overlap, but you have to consider that this is only looking at the 23, 24 focal species. And this is everything else, yeah, that we have not been actively monitoring. But okay, it's, you know, during the same season, different times, maybe there's a little bit like micro differences in the climate when you were collecting. So why didn't we collect everything in the metabarcoding? It's open question. But once we took the 14 years of light trap data, so that's about 1,200 light trap data that the Arthropod initiative has, and we compare it to our metabarcoding, and just in one season or 2 seasons of metabarcoding, we get about half the total species that have been monitored by the Arthropod program. And okay, the other half we don't get because this has been going for 14 years, right? There can be year-to-year variation that we just don't capture in the one event. So with this in mind, I hope I have convinced you that we have enough foundation to start using metabarcoding as a monitoring tool for what is out in the forest. Okay, I should have explained. You're probably asking, okay, Daniel, if these 3,000 and some species were not part of the monitoring program, why do you have those sequences, right? How do you know? Well, if you think that we can build databases of what everyone else has sequenced, so that's from both, a lot of them come from Costa Rica with Dan Jansen and Guanacaste sequencing a bunch of different animals. And then, you know, these are community-wide effort from the whole world. So we are standing in the shoulders of barcoding giants. So now, okay, for the Sena seat, we have these 7 different traps. Well, I tried to make like really loose comparisons or categories, let's say. So flight intercept, we had the classic Malaise trap. I think this is used the world all over. And the poly trap, which is basically a flight intercept. The animals kind of crash into this panel that you can't see. They either fly up and come to this collection vial, or they bounce down and they fall into this other collection vial. Yeah, this is the flight intercept. We have Winkler and Berles Tuliran funnels. These target soil or leaf litter fauna. For the Winkler, you just collect like the top debris, put it into some sieve, and you shake it really hard, and everything falls into some collection pocket. Then you can hang those traps. The Berles Tuliran funnel is similar, but then you have like a light bulb on top that will start warming the soil that you collected. You actually collect topsoil. It warms the soil, it dries it up. So every critter that lives in there tries to crawl down to where it's a bit more humid, and they fall into a ethanol-filled vial. Everything is killed with ethanol. Light trap is a little bit on its own because it's actually a baiting trap, right? It's a big light in the forest that's attracting all nocturnal animals which are attracted to light. But it works as an intercept trap. They fly, they crash, and then they fall into a trap of ethanol. We have the pitfall trap. So this one, you have to go into the field and dig a hole, and we put like a little party cup, more or less. It's very low budget. It's very easy to make, and it's just a dish to prevent the rain. And beating, which is like a very old method of collection. We just go with a sheet and you kind of hit the vegetation, and whatever animal falls into your canvas. And in the bottom, yes, an ethanol trap. All traps have like advantages and disadvantages. Particularly these two, and actually the litter fall, they collect a lot of debris from the forest, like leaves and soil. And so then you have extra steps in the laboratory to clean your ethanol before you can do your extractions. So they add some labor. Probably the cleanest ones are the Malaise, the light trap, because you just have like a sealed container and everything falls into it. And I wasn't here for the poly trap, but I know that the top trap was super clean, but the bottom trap was not. You can already even see it here. And that's because like rain and debris and everything falls into the traps. We will discuss about this too. But yes, we will get into it right now. Eve showed us already the distribution of orders, but along the plots, but now I can show you with different trap types, right? I think the most immediate thing that calls the attention is the light trap, because moths, but because of course they are attracted to light, so it works as intended, but it collects a lot of other things that are not the target species. But more or less, it's Malay trap captures a lot of flying insects. That's no surprises there. They all collect a different degree of diversity. This number of bins and each trap type. So you can see that the pitfall is not very diverse. Like it's quite not a lot of insects, and the beating also. And okay, not groundbreaking results here. We all know that different traps target different groups, but we're talking about 10,000 species that are falling into these traps. I think that is significant. And okay, we have 2 seasons, wet and dry, and you can already start easily to compare that each season. I didn't put which one is wet and dry. These are all like preliminary as of yesterday, okay? We just received the total data and we've been working on that. But okay, there is differences between wet and dry. I already knew we could find that through the light trap study, but now we can see that for all the traps, it's a similar story. But now, if we combine the methods into like these smaller categories that I made, flying, so it's polytrap and wing and Malays, hand is just the beating, light trap on its own, and soil is pitfall, winkler, and burlesque. You can start to see that the forest structure kind of, or the target structure can actually be conglomerated. And when you start looking at what is the overlap between different methods, okay, this is in those 4 fake categories, because a 7-category Venn diagram, I was gonna put a picture here, but you should look it online. It's impossible to interpret, but it looks very psychedelic. But you can see that, okay, now let's pretend funding is a huge issue in science. You don't have unlimited funding, and it's probably rare that you will get funding to do, again, 7 different protocols for monitoring insects. I think this, Senacit took a gamble, a big gamble, which we are now providing good results, I think. But if your funding is a bit more limited and you want to compare soil versus flight, okay, either one of those traps are going to give you a good number. This is the total bins for each trap type and their overlap. So it's a bit, this table is the same as here, but a little bit more broken down into what the beating, what is the overlap with soil and flying, etc. And with this, you can start also to see, this is the same graph as before, but now with those combined categories. But this will allow us now to choose what are the best methods or cost-effective methods to actually monitor tropical forests. So Malay trap, okay, in total it's 4,600 beans, polytrap 4,500. They both have unique beans, okay? They're not sharing everything, but you don't get huge difference in numbers. So which one is more efficient to set up? Which one is less maintenance? For example, the polytrap is a little bit heavy to carry, but you can leave it there for a long time. You just gotta go and change the vials. Malays also, but it's targeting only things that will fly underground and go up. Things that fly, hit, and fall down, they're not being captured. So what is the ecology of the species and the groups that you're interested on will define what trap you would like to use. For soil, kind of the same. Interesting that winkler and burlesque, more or less the same. Okay, I know 300 species doesn't seem like the same, but we're talking about thousands now, so maybe it's not too different. But the winkler produces a lot cleaner traps than the burlesque, because the burlesque actually has the soil, and there are extra steps in the lab that can be costly. So the funny thing is that the pitfall traps, although they don't vary too much between the traps themselves, it captures a lot of insects too. But a lot of them were spiders. So if it's not your target group, you're actually looking at soil fauna, well, you can choose one of those two. And then I think maybe the Light trap is ideal, but again, I think one of the biggest disadvantages of the light trap is that it's a bait trap in a way, because you have the light and it's attracting the insects. It's not passive, right? Like you're actually calling the insects to that trap. Ethanol kind of does the same effect, but to a smaller degree than a giant light in the forest. You have to carry batteries, you have to recharge those batteries. This one is probably one of the heaviest ones to actually bring into the forest, but it's super effective. It's the one that collected the most diversity. Beating doesn't get that many beans, but it's probably one of the most fun methods to use because you actually see all the animals instead of just like a collection of stuff. You go, you hit, and there's different ways to standardize all of these methods. I think the point to make, besides the funding, is that you really need to think about the ecology of the target group that you're looking for, and then you can decide on which ones. And I know it's not like the best recommendation. I wish I could say, oh, you take the winkler and you take the light trap and you have all diversity of the forest. But with more analysis, I think we can narrow down these recommendations a little bit more. And with that, I just want to thank Eve a lot for allowing me to be part of this study, and everyone else, yeah, for listening. Just in time.
We already past time, so maybe we can call everybody for a coffee break. Maybe you can speak to
We will have coffee break.
for half an hour.
Half hour, and we'll be back.
10:30.
So 10:30, we'll be back.